Hi, I'm Geoffrey Chariot. I'm going to make a reproductive system presentation about the regulation of sperm maturation in the germinal epithelium. So to start us off, let's go through the basic anatomy of the male reproductive system. The male reproductive system is divided into three main anatomical areas. So the first part is the internal genitalia, which includes the accessory glands and ducts. So the internal genitalia includes prostate glands, seminal vesicles. So seminal vesicles, the prostate gland is here, seminal vesicle is just above it over here. The bulbourethral gland is this small gland over here, and it's also called a Cowper's glands in some other literature. And the second part of the male reproductive system include the external genitalia, which basically include the scrotum and the penis. The penis is all the structure, the physical structure, all of it. And then the scrotum is this bag, or this bag that covers all the testes over here. The third part of the male reproductive system is the testis, which is basically the gonad, the main reproductive structure in humans and the male humans and animals. It is within these testes that we have the seminiferous tubule. Seminiferous tubules are these coiled structures over here, and those are the site of spermatogenesis, the process of sperm creation. So the testes contain supercoiled seminiferous tubules, which are the site of spermatogenesis. We can see from this cross-section structure of the testis tissue, all these highly coiled structures, these are the seminiferous tubules. The seminiferous tubule has two types of cells, which are the spermatogonia. Spermatogonia is over here on this basement membrane. They are attached to the basement membrane over here. And these are the progenitor cells for the development of sperms. So these are the ones that undergo various forms of cellular division, cell division processes, mitosis and meiosis, just to give you a preview of that, to become sperms. The Sertoli cells are these big structures over here. So Sertoli cells provide nourishment. They provide nourishment for the development of sperms by making hormones inhibin, activin, other growth factors, enzymes, and ABP. This is just a structure with all showing all the Sertoli cells, the spermatogonia, the basement membrane on the outside. All the just a simple cellular division process here. We will go in depth in the next slide. So the process of spermatogenesis, sperm formation. So spermatogonia, which is the germ cell found clustered near the basal ends of the Sertoli cells inside basal lamina of seminiferous tubules. It's found over here near the basement, the basal lamina of the seminiferous tubule, and as it develops, it moves inside towards the lumen inside here. So spermatogonia undergo mitosis. Spermatogonia undergo mitosis to produce primary spermatocyte, which is a diploid cell, which are diploid. So spermatogonia here undergo division to produce primary spermatocyte here. The primary spermatocyte undergo a different form of cell division called meiosis to produce secondary spermatocyte, which is haploid. This is happening over here. And then the secondary spermatocyte undergo meiosis to produce spermatids. So after the secondary spermatocyte is formed, it undergoes the second part of meiosis to produce spermatids. The spermatids remain in the apical membrane of Sertoli cells until they mature into sperms. So they are not ready to be sperms yet, so they hang around the Sertoli cells. And we must remember that as this arrow is showing, I'm just trying to show that as sperms differentiate into sperms, they move inward towards that tubule lumen, the inside over here. The entire process of spermatogenesis takes 64 days until sperms are released to the lumen of seminiferous tubule. We must also remember that by the anatomy-wise, that the process of spermatogenesis takes part within the kind of outside the body temperature. That's why the testis hangs outside the body. So it's the temperature of, or the optimum optimum temperature for sperm generation is a little bit slightly lower than that of the body. To continue with the process of spermatogenesis, after the sperms are released, they are not mature and cannot spin or are immotile. So they move towards the epididymis for more maturity and with the help of protein secretion from epididymal cells. So they move here for a little bit more of maturity and nutritious nourishment. Okay. As we can see, this is the epididymis over here, the vas deferens. Tunica albumina is this shell over here that encloses the seminiferous tubules over here. Okay. The role of gonadotrophins and testosterone in spermatogenesis. Here we're looking at the role of various hormones in facilitating the process of spermatogenesis. So let's start with gonadotrophin releasing hormone GnRH. So GnRH promotes release of gonadotrophins, which are LH and FSH, from the anterior pituitary. So LH is luteinizing hormone and FSH is follicle stimulating hormone. The luteinizing hormone, LH in short, its job is to act on Leydig cells to produce testosterone. The Leydig cells are the cells that produce testosterone. Okay, the FSH, follicle stimulating hormone, stimulates the Sertoli cells, which are the somatic cells involved in spermatogenesis to begin spermatogenesis. So it basically initiates the process of spermatogenesis. It also stimulates the production of proteins such as inhibin and ABP. Just a side note here, since luteinizing hormone stimulates Leydig cells to produce testosterone, there is a negative feedback system exists where testosterone also feeds back to inhibit LH and gonadotrophic releasing hormone. So it's a negative feedback that controls the production of these. Whenever we have, whenever the Leydig cells produce enough testosterone and LH, there is a regulatory mechanism that uses a negative feedback system.